What is Asfarviridae? Asfarviridae is a family of viruses named after African swine fever and related viruses. It contains only one genus, Asfivirus, and one known member, the African swine fever virus. African swine fever virus is the causative agent of African swine fever, a highly contagious and often fatal disease affecting domestic and wild pigs. This virus poses a significant threat to the global swine industry due to its high mortality rates and the lack of an effective vaccine. Structurally, African swine fever virus is an enveloped virus with a complex icosahedral capsid. It measures approximately 175 to 250 nanometers in size, with its capsid consisting of at least 1,892 capsomers and a diameter of 180 nanometers. The virus contains a large, linear double-stranded DNA genome ranging from 170 to 190 kilobase pairs. This genome encodes up to 200 proteins, and many of which play roles in viral replication, immune invasion, and host infection. Unlike most DNA viruses, African swine fever viruses primarily replicates in the cytoplasm of infected cells. Although the nucleus is necessary for viral DNA synthesis, newly formed virions are released through either budding or cell lysis. One of African swine fever virus defining characteristics is its variable virulence. Some strains cause near 100% mortality in pigs, leading to severe outbreaks, while others result in milder or even asymptomatic infections. The virus is primarily transmitted through direct contact between infected and healthy pigs, as well as through contaminated feed, equipment, and clothing. However, African swine fever virus is also unique as the only known DNA arbovirus, meaning it is transmitted by arthropods. In this case, soft ticks of the Ornithodorus genus, these ticks serve as both biological vectors and reservoir, further complicating efforts to control the disease.
African swine fever virus was initially classified under the family Iridoviridae, a group of viruses that primarily infect insects, fish, and amphibians. Although African swine fever virus shares some morphological features with iridoviruses, researchers later discovered major differences in its DNA structure and replication strategy. These findings led to African swine fever virus being reclassified as the only member of a new family, Asfarviridae. The virus also exhibits genetic similarities to the other large DNA viruses, particularly pox viruses, though their evolutionary connections remains distant. African swine fever virus genome structure resembles linear plasmid found in bacteria of the genus Borrelia, a cause of relapsing fever in Africa. Both African swine fever virus and Borrelia share the same thick vector, the Ornithodorus pubata. Some scientists have suggested that African swine fever virus and Borrelia may share a common ancestry or that horizontal gene transfer has occurred between them over time.
African swine fever virus is the sole member of the Asfarviridae family and the Asfarvirus genus, making its classification relatively straightforward. However, DNA analysis reveals that African swine fever virus isolates can be divided into five genetic groups, while European and American isolates belong to a single group. African isolates exhibit much greater genetic diversity. This suggests that African swine fever virus has circulated in Africa for a longer period, allowing for significant genetic divergence over time. Structurally, African swine fever virus is a large, enveloped DNA virus with a highly complex genome. It is the only known DNA arbovirus, meaning it is transmitted by arthropods. And the genome contains unique multi-gene families, and the virus strains vary in virulence, ranging from highly lethal to subclinical infections. These variations in severity complicates disease control efforts. Genetically, African swine fever virus is a highly diverse virus, and one of the key genes used for genotyping is P72, or also known as P73, which helps differentiate between strains. However, despite extensive research, the full genomic diversity of African swine fever virus remains incompletely understood. Additionally, African swine fever virus contains unique multi-gene families, which play a role in its adaptability and ability to evade the immune system. The African swine fever virus, measuring around 200 nanometer in diameter, its structure includes a complex icosahedral capsid, which is arranged in hexagonal units, each appearing as a hexagonal prism with a central hole. Beneath the capsid, the virion has internal lipid layers surrounding a nucleocapsid core. African swine fever virus possesses a linear double-stranded DNA genome that varies between 170 and 190 kilobase pair, depending on the strain. The genome has covalently close ends along with the inverted terminal repeats and hairpin loops, which contribute to its structural integrity. Within the genome, there are approximately 150 open reading frames, which are closely spaced and transcribed from both DNA strands. African swine fever virus virions contains more than 50 proteins, including enzymes required for early mRNA transcription and processing. This allows the virus to immediately begin replication upon infecting a host cell, making it highly efficient at establishing infection. One of the key characteristics of African swine fever virus is its stability in the environment. Although it is thermolabile and can be inactivated by lipid solvents, it is highly resistant to extreme pH levels. The virus can survive for several hours in acidic or highly alkaline conditions. More importantly, African swine fever virus can persist for months or even years in refrigerated pork products, which makes contaminated meat a major transmission risk. These unique virions properties contribute to the high resilience and persistence of African swine fever virus, making it a serious threat to global swine population and a challenge for disease control measures. In this figure, it shows the African swine fever virus, a large and complex virus using electron microscopy. The figure is composed of 4 panels, each showcasing a different aspect of the virus structure. The image A presents an intact African swine fever virus virion and the complete infectious form of the virus. Negative staining, a technique where the background is stained rather than the virus itself, is employed. This reveals the overall structure, highlighting the hexagonal outline of the capsid, and the protein shell containing the viral genome, enclosed within the viral envelope. For the image B and C, display damaged African swine fever virus capsids, also visualized using negative staining. The damage, likely an artifact of the preparation process, exposes the intricate arrangement of capsomeres, the protein subunits that make up the capsid. The high degree of order and the sheer number of capsomeres, estimated to be between 1,892 and 2,172, are evident. This emphasizes the structural complexity of the virus. For the image D, it shows a thin section of African swine fever virus virions. These techniques involve slicing the sample into extremely thin sections, enabling visualizations of the internal organization. Multiple layers surrounding the viral core are visible, representing different components of the virus particle, including the core containing the genetic material, proteins involved in replication, and the outer envelope crucial for host cell entry.
For the explanation of virus replication in African swine fever virus, it replicates mainly in the cytoplasm of infected cells, specifically targeting the monocytes and macrophages. Why primarily target the monocytes and macrophages? Because these immune cells plays an important role in the pig's immune response. Therefore, the African swine fever virus disables the pig's immune defenses where it spreads systemically, making it highly lethal and difficult to control. For the mechanism of virus replication in African swine fever virus, number 1 is infected cells and replication in monocytes and macrophages. Primarily infect monocytes and macrophages, which are the types of white blood cells in pigs. These cells are where the virus first replicates, but after the virus adapts to different conditions, it can also replicate in certain mammalian cell lines. Number 2 is entry and virus binding. African swine fever virus enters the cells through a process called receptor-mediated endocytosis. This is when the virus attaches to specific receptors on the surface of the cell and gets engulfed into the cell. The virus uses its protein, particularly the P72 and P54, that helps the virus bind to the cell surface, and P30 is involved in bringing the virus inside the cell once it attaches. Number 3 is viral DNA synthesis and replication. Once inside the cell, the virus releases its genomic DNA into the cytoplasm, which is the liquid part of the cell. This DNA contains everything the virus needs to replicate and make new virus particles. Like pox viruses, African swine fever virus brings along the machinery required for transcription and replication. The virus uses its own RNA polymerase to transcribe the viral DNA. This is similar to how pox viruses do it. The viral DNA replication process begins with the virus using its original DNA to create more copies of itself. These new copies then serve as templates for the synthesis of even more viral DNA. Number 4 is virus assembly and release. Later in the infection, the virus begins to assemble in the cytoplasm. It forms the paracrystalline arrays, which are basically tightly packed structures of virus particles. The infected cells develop projections similar to tiny finger-like structures called the microvilli. These projections allow the virus particles to bud out from the cell. However, African swine fever virus does not need to acquire an envelope around its virus particle to be infectious, which is different from some viruses that require an envelope for infectivity. For the initial outbreaks of African swine fever virus, before 1957, African swine fever virus was thought to be limited to sub-Saharan Africa. In 1957, African swine fever virus spread beyond Africa for the first time, reaching the Iberian Peninsula. This marked the beginning of its expansion beyond Africa. Throughout the 1970s, sporadic outbreaks, meaning the disease appears infrequently in isolated areas, were reported in Caribbean islands including the Cuba and the Dominican Republic. By the 1980s, African swine fever virus appeared in the European countries such as France and Belgium. Since 2007, African swine fever virus has spread further, reaching countries in Eastern Europe, including Georgia, Armenia, Azerbaijan, and Russia. Number 2 is endemic regions. Despite its spread, African swine fever virus remains exotic in some areas, including the sub-Saharan Africa and Sardinia. In Sardinia, African swine fever virus continues to circulate due to the presence of wild boar and extensive pig farming. These wild boar populations and farming practices help sustain the virus in the region. Number 3 is host of African swine fever virus. This primarily infects the domestic pigs, Sus scrofa domesticus, and other wild members of the Suidae family, such as the warthogs, Potamocherus aethiopicus, bush pigs, Potamocherus porcus, and wild boar, Sus scrofa ferus. These attempts to infect other animals have been unsuccessful, meaning the African swine fever virus appears to be specialized for this species only. Vector and potential tick origins. It's hypothesized that African swine fever virus might have originally been a virus of ticks. In Africa, the virus has been isolated from the soft tick species Ornithotodorus maubata, which is commonly found in warthog burrows. When African swine fever virus was considered confined to sub-Saharan Africa, it was believed that the virus spread was tied to its natural cycle involving the argacid ticks and wild swine. However, the virus has occasionally spread beyond this range. In Europe and other species of soft tick, Ornithotodorus erraticus may serve as a vector for African swine fever virus, making the virus spread further. Properties of Asfarviruses and Iridoviruses. Asfarviruses have an outer lipid membrane called the envelope, which helps the virus enters the host cells. For the size, the virus is approximately 200 nanometer in diameter, and the icosahedral capsid is about 180 nanometer in diameter. An icosahedral capsid has a symmetrical 20-sided shape, which is common for many viruses. The genome type of Asfarviruses has a single molecule of linear double-stranded DNA, meaning its genetic material is made up of two complementary strands of DNA arranged in a single straight line. The size of the genome is around 170 to 190 kilobase pairs in length, which is relatively large for a virus. The structure of the DNA has a covalently close, meaning they are connected to form a continuous loop. It also has a inverted terminal repeats, meaning the repeated DNA sequences at both ends in reverse order, and hairpin loops, meaning the DNA structures that forms loops at the ends. For the protein encoding, Asfarviruses encodes about 150 proteins, with over 50 of them being part of the virion. These proteins play various roles in the virus structures, replication, and interaction with host cells. The comparison with vertebrate in iridoviruses. Vertebrate iridoviruses have a similar virion structure to asfarviruses. They also have a linear double-stranded DNA genome with a size range of 140 to 200 kilobase pairs and can encode up to 200 proteins. The iridovirus genome is permuted circularly, which means the genetic sequence is mixed up in a circular pattern. Like as for viruses, it is also a terminally redundant ends, meaning the repeated sequences both ends and methylated bases where certain DNA bases are chemically modified. For the replication and assembly, the nucleus of the host cell is involved in the DNA replication. This is where the viral genome is copied. After replication, the late functions of the virus, such as the synthesis of new viral proteins and assembling of the virus particles, and virion assembly occur in the cytoplasm of the host cell.
Clinical features. The Asfarviridae family, which includes the African swine fever virus, is characterized by specific clinical features and epidemiological patterns. African swine fever in susceptible swine is a severe hemorrhagic disease with high mortality. After an incubation period of 5 to 15 days, swine develop a fever of 40.5 to 42 degrees Celsius that lasts about 4 days. Starting 1 to 2 days after the onset of fever, there is inappetence, diarrhea, incoordination, and prostration. Some swine may show dyspnea, vomiting, nasal and conjunctival discharge, reddening or cyanosis of the ears and snout, and hemorrhages from the nose and anus. Pregnant sows often abort. Mortality is often 100% in domestic swine, with death occurring within 1 to 3 days after the onset of fever. Initially, the disease is severe and fatal, but it can diminish quickly until cases are predominantly subclinical and persistent. Infected adult warthogs do not develop clinical disease. Swine that survive acute infection may become persistently infected and may show no clinical signs or be chronically diseased. In acutely fatal cases, gross lesions are most prominent in the lymphoid and vascular systems, with widespread hemorrhages. Visceral lymph nodes may resemble blood clots, and there is marked petechiation of all serous surfaces, lymph nodes, epicardium and endocardium, renal cortex, and bladder. Edema and congestion of the colon and lungs are also observed. The spleen is often large and friable, with petechial hemorrhages in the cortex of the kidney. Chronic disease is characterized by cutaneous ulcers, pneumonia, pericarditis, pleuritis, and arthritis. These clinical signs can be similar to those of other diseases, making laboratory confirmation is essential. The epidemiology of African swine fever virus involves two distinct patterns of transmission: the sylvatic cycle and the domestic cycle. Sylvatic cycle occurs in southern and eastern Africa, where the virus is maintained through asymptomatic infections in wild pigs, specifically warthogs and bush pigs, and argacid ticks, Ornithodoros genus. Ticks are biological vectors. The virus replicates in their gut, infecting the reproductive system, leading through transovarial and venereal transmission, as well as transstadial transmission. Infected ticks can transmit the disease to swine at each blood meal and can live for several years. Young warthogs develop viremia after primary infection, sufficient to infect ticks, while older warthogs are persistently infected but seldom viremic. The primary source of the virus in African swine fever is epizootics in southern and eastern Africa, are infected ticks that are transported by live warthogs on their carcasses. Domestic cycle primary outbreaks in domestic swine in Africa likely result from the bite of an infected tick or through eating tissues of acutely infected warthogs. High virus titers are present in nasopharyngeal excretions and feces during acute disease. The disease spreads rapidly through contact and via aerosol within buildings, and through mechanical spread by people, vehicles, and fomites. International spread has been linked to feeding uncooked meat scraps from infected swine. Other epidemiological considerations. African swine fever was initially considered a disease of sub Saharan Africa until 1957, when it spread to the Iberian Peninsula. Sporadic outbreaks occurred in the 1970s in the Caribbean islands and in the 1980s in Europe. Since 2007, the virus has spread throughout portions of Georgia, Armenia, Azerbaijan, and Russia. The disease remains enzootic in sub-Saharan Africa and Sardinia. The virus may have originated in ticks but have spread beyond its traditional range into areas where Ornithodoros erraticus can be a vector. Several soft tick species found in association with domestic and feral swine in the Western Hemisphere have been shown to be capable of transmitting the virus. Although there is no evidence they became infected during the epizootics in the Caribbean islands and South America. The pathogenesis of Asfarviridae, specifically the African swine fever virus or ASFV, involves several key aspects in domestic swine. Here's a breakdown. Impact on blood cells. ASF infection in domestic swine leads to leukopenia, lymphopenia, and thrombocytopenia, as well as apoptosis of both lymphocytes and mononuclear phagocytic cells. Macrophage interaction. ASF ability to cause cytopathology in macrophages is crucial to its virulence. In infected macrophages, the virus inhibits the expression of pro-inflammatory cytokines such as tissue necrosis factor or TNF, type 1 interferon, and interleukin 8, while inducing expression of transforming growth factor beta. However, increased expression of TNF has also been observed in some cases. Immune evasion. The virus uses a novel strategy to evade the immune system by employing the viral gene A238L. It is a non-structural protein found in the African swine fever, which encodes a protein similar to an inhibitor of cellular transcription factor, the NFKB or nuclear factor kappa B. This protein inhibits NFKB activation, downregulating the expression of antiviral cytokines, viral proteins, and immune modulation. Additional ASF virus proteins, such as 8DR, which is a viral homologue of cellular CD2, are involved in T lymphocyte activation and mediation of hemadsorption in infected cells. Replication and dissemination. If the virus enters through the respiratory tract, it is initially replicates in the pharyngeal tonsils and lymph nodes before spreading throughout body via primary viremia, where virions are associated with both erythrocytes and leukocytes. A generalized infection follows with high virus titers in blood and tissues. For the pathology of Asfarviridae, it involves acute infections, chronic infections, and persistent infection. For the acute infections, in acute, fatal cases, lesions are prominent in the lymphoid and vascular systems. There are widespread hemorrhages, and visceral lymph nodes may resemble blood clots. Petechiae are found on serous surfaces, lymph nodes, epicardium, endocardium, renal cortex, and bladder. Additionally, edema and congestion of the colon and lungs can occur. The spleen is often enlarged and friable, and hemorrhages are found in the kidney cortex. For chronic infections, chronic disease is marked by cutaneous ulcers, pneumonia, pericarditis, pleuritis, and arthritis. Lastly is the persistent infection. Swine that survive the acute infection may remain persistently infected, sometimes without showing clinical signs. The virus can be detected in tissues for more than a year after exposure.
Good day, everyone. I'm Christine Pizzas Quito Valenzuela, and I'll be discussing how we diagnose African swine fever or ASF. Diagnosing ASF is crucial because its symptoms resemble other swine diseases, making early detection challenging but essential to prevent outbreaks. Its clinical diagnosis is ASF can look a lot like other diseases, such as erysipelas, acute salmonellosis, and classical swine fever or hog cholera. The key signs to watch for include are high fever, widespread hemorrhages, and high mortality rates. In chronic ASF cases, symptoms can be more variable, making clinical diagnosis even more difficult. That's why laboratory testing is a must. To confirm ASF, we collect and test samples from affected pigs including their blood, spleen, kidney, lymph nodes, and tonsils. These samples are used for virus isolation, antigen detection, or PCR testing to identify the SF virus. The key diagnostic methods are: number 1, virus isolation. Virus is grown in swine bone marrow or blood cultures. Number 2, hemadsorption or clumping of red blood cells is observed. Third one is cytopathic effects or cell damage. It appears in a few days. So the last one for the ASF diagnosis is antigen detection. As you can see below in the picture, it is immunofluorescence staining. It highlights ASF virus in tissue smears. Next, immunodiffusion. It uses tissue suspensions to detect viral proteins. Last one is enzyme immunoassay or EIA. It is a reliable method for detecting ASF antigens. In conclusion, because ASF closely resembles other swine diseases, combining clinical evaluation with laboratory tests is essential. Early detection and rapid response are the keys to controlling the spread and protecting the swine industry from major losses. So these are pictures of lesions of acute African swine fever. In the first picture or letter A picture, it is a subcutaneous hemorrhages in the ear. These indicate widespread vascular damage and bleeding under the skin, a common sign of severe ASF infection. Next is the picture B. It shows splenomegaly, an abnormal enlargement of the spleen due to intense viral replication and immune response making it dark and friable. Now, I'll be talking about the immunity, prevention, and control of ASF. When it comes to immunity, both humoral and cellular responses play a role in protecting pigs from ASF. A key component of this immune defense is the CD8 plus lymphocyte, which helps fight off the virus. Studies have shown that antibodies can protect pigs from severe infection, but unfortunately, they aren't enough to fully stop the disease. Even though antibodies target viral proteins like P30, P54, and P72, they don't provide complete protection on their own. Controlling ASF is extremely challenging for several reasons. First, there is no effective vaccine to prevent infection. Second, the virus can spread through contaminated fresh meat and some cured pork products. Third, some pigs can harbor the virus long term, making it difficult to eliminate. Fourth, ASF symptoms can be mistaken for other diseases like classical swine fever, leading to delays in detection. Last is, in some regions, soft ticks act as carriers, spreading the disease further. In the next slide, you'll be seeing a picture of a tick, specifically the ornithodoros ticks. Domestic pig farming can still be done safely if proper management practices are followed. First, avoid feeding uncooked food scraps to pigs as they may contain the virus. Second, prevent tick infestations and keep pigs away from warthogs using double fencing with a wire mesh that extends underground to stop burrowing animals. For ASF-free countries, strict regulations help prevent outbreaks. First, banning the import of live pigs and pork products from infected areas. Second, ensuring the proper disposal of food waste from international ships and planes so contaminated scraps don't reach local pigs. With regards to responding to outbreaks, if ASF is detected in a country for the first time, quick action is essential. The highly virulent forms of the virus cause severe disease and mass pig deaths, which makes detection easier. However, less virulent strains can go unnoticed, allowing the virus to spread silently before anyone realizes there's a problem. Once ASF is confirmed in a country, the only way to eradicate it is through strict measures. First, slaughtering infected pigs and those in contact with them to stop further spread. Second, proper disposal of carcasses, preferably by burning to destroy the virus. Third, restricting pig movement between farms to prevent transmission. Fourth, banning food waste feeding to eliminate another source of infection. Fifth, spraying acaricides in tick-infested areas to control the vector. Last, restocking farms only after ensuring that test pigs or sentinel swine remain uninfected. In sub-Saharan Africa, ASF is particularly hard to control because the virus circulates naturally between warthogs and soft ticks. Breaking this cycle is nearly impossible. So in conclusion, ASF remains one of the biggest threats to the swine industry worldwide. Since no vaccine exists, prevention relies on strict biosecurity, early detection, and rapid response. The key to controlling ASF is stopping its spread before it becomes uncontrollable. Thank you.
What is Iridoviridae? The Iridoviridae family is a large and diverse group of viruses known to infect a wide range of hosts, including arthropods, fish, amphibians, and reptiles. Among the most significant genera are Ranavirus, Megalocytivirus, and Lymphocystivirus, each of which impacts aquatic species differently. Both Ranavirus and Megalocytivirus are responsible for severe systemic lethal diseases in fish, leading to high mortality rates. Meanwhile, Lymphocystivirus is known for causing tumor-like skin lesions in fish populations. Beyond their impact on fish, ranaviruses have been identified as a potential contributor to the global decline of amphibian population, posing a serious threat to biodiversity. One of the key characteristics of these viruses is their ability to persist long-term in fish and amphibians, even after an acute infection or seemingly mild illness. The virus can remain in the host, potentially leading to future outbreaks or transmissions to other animals. Iridoviruses belong to the family Iridoviridae are DNA viruses with a size range of 120 to 200 nanometer, but some species can be even larger. The virus structure is characterized by icosahedral symmetry with a virus core and outer capsid, separated by an internal lipid membrane. These virions contain up to 36 proteins, and while a viral envelope is found on virions that bud from infected cells, it is not essential for their ineffectivity. The genome of iridoviruses consists of a single, linear double-stranded DNA molecule, with a length between 140 to 200 kilobase pairs, encoding 100 to 200 proteins. The genome also exhibits circular permutations and terminal redundancy, and in certain species, genomic methylation occurs, involving up to 20% of cytosine residues. The family is subdivided into five genera: Iridovirus, Chloriridovirus, Ranavirus, Megalocytivirus, and Lymphocystivirus. These viruses have significant pathogenic importance, causing disease in commercial fish and leading to mortality in both amphibians and reptiles. Some arthropods-related genera like Iridovirus and Chloriridovirus have also been found in lizards and scorpions, suggesting potential transmissions via insect prey. The replication cycle of iridoviruses is similar to that of the African swine fever virus. However, iridoviruses do not encode their own RNA polymerase and instead use cellular RNA polymerase II to synthesize viral mRNA. Late in the infection, paracrystalline arrays of virions are produced in the cytoplasm. The virions may bud through microvillus-like projections of infected cells, but the viral envelope is not required for the virus to be infectious. The virus particles are released after the lysis of the infected cells. In this figure, it presents a view of iridoviruses, a group of large complex viruses that infect a wide range of invertebrates and in some cases vertebrates. The top left image, this image showcases the outer shell of invertebrate iridescent virus 2 or the IIV2. The intricate icosahedral structure is evident, highlighting the precise arrangement of the protein subunits that form the capsid. The capsid provides protection for the viral genome and plays a crucial role in host cell recognition and entry. The top right image, a schematic diagram illustrates a cross-sections of an iridovirus particle. The diagram highlights key structural components including the capsomeres, the individual protein units comprising the capsid, and the transmembrane proteins embedded within the lipid bilayer of the viral envelope are also shown. These proteins are essential for membrane fusion and entry into the host cell. The diagram depicts the internal filamentous nucleoprotein core, which contains the viral genome and associated proteins. The bottom left image, a transmission electron micrograph of a fathead minnow cell infected with European catfish virus, provides a glimpse into the viral replication process. The image identifies the nucleus, labeled as NU, and a prominent virus inclusion body, labeled as BIB, a region within the cytoplasm where viral assembly takes place. A paracrystalline array of non-enveloped virus particles, the arrows, indicates the high concentrations of virions within the infected cell. Incomplete nucleocapsids, which is the arrowheads, suggest different stages of viral development. The cytoplasm and mitochondrion are also labeled, providing cellular context. The transmission electron micrograph depicts frog virus 3, the FV3, budding from the plasma membrane of an infected cells, and the arrows and the arrowheads highlight the viral envelope acquired during this budding process. This envelope is derived from the host cell membrane and further modified with viral proteins.
Taxonomy of family Iridoviridae. Number 1, the genus Iridovirus has a virus species invertebrate iridescent virus 6. 1, 2, 9, 16, 21, 22, 23, 24, 29, 30, and 31. This genus includes the viruses that infect invertebrates like arthropods and other marine species, often causing iridescent lesions in their hosts. Number 2, genus Chloriridovirus has a virus species called invertebrate iridescent virus 3. Chloriridoviruses also affects invertebrates, causing iridescent or color-changing effects in their host organisms. Number 3 genus is Ranavirus. The virus species are Frogvirus 3, also known as tadpole edema virus or tiger frog virus, Ambystoma tigrinum virus, also known as Regina varana virus, epizootic hematopoietic necrosis virus, European catfish virus, also known as European shitfish virus, Santi-Cooper virus, also known as largemouth bass virus, Dr. Fish virus, or Guppy virus 6, Singapore grouper iridovirus. Ranaviruses are primarily known to infect amphibians and fish, including the diseases like tadpole edema and epizootic hematopoietic necrosis. Number 4 genus is Megalocytivirus. The virus species includes the infectious spleen and kidney necrosis virus, also known as Red Sea Bream iridovirus, African Lampi iridovirus, orange-spotted grouper iridovirus, and rock bream iridovirus. Megalocytiviruses infect marine and freshwater fish, causing a systemic diseases that can lead to necrosis in the spleen and kidneys. Number 5 genus is Lymphocystivirus. The virus species includes the Lymphocytis disease virus 1 and number 2. These viruses cause lymphocytis disease, which leads to the formation of tumor-like cysts in fish, particularly in marine fish species. Number 6 is unclassified virus. The white sturgeon iridovirus. This virus is not classified into a specific genus within the family Iridoviridae. This virus infects the white sturgeon, a species of fish, and is characterized by severe systemic infections. The initial detection and spread of frog virus 3 was first detected in the 1960s from leopard frogs in the eastern United States, which was linked to renal sarcoidomas, which is the kidney tumors in frogs, which were later traced to an oncogenic herpes virus, which is the ranid herpes virus 1. Initially, ranaviruses like frog virus 3 were thought to be relatively benign and not a major threat to amphibians. However, by the mid-1980s, it became clear that ranaviruses were causing severe disease outbreaks in wild amphibians across North America, Europe, and Asia. The symptoms and signs that infect tadpoles and frogs may show localized hemorrhage, bleeding under the skin, and ulceration. Severe systemic disease with symptoms like edema, hemorrhage, and necrosis in multiple organs. Subclinical infections can occur where infected amphibians may show no obvious symptoms but still harbor the virus, especially in the kidney tissues where the virus can persist inside the macrophages. Ambystoma tigrinum virus is a rhinovirus that infects salamanders in western North America, causing high mortality up to 90% in both larval and adult salamanders, particularly during late summer and early autumn. Infected salamanders may exhibit necrosis and hemorrhage in organs like spleen, liver, kidneys, and gastrointestinal tract. Skin sloughing, development of skin polyps, and inflammatory exudate, which is the fluid, and discharge from the vent, which is the anus. Temperature plays a significant role in the infection severity. Salamanders infected at 26 degrees Celsius have a higher survival rate, while at 18 degrees Celsius the infection is much more lethal. The vertical transmission of the virus from infected adult salamanders to their eggs is unclear, meaning it's not well understood whether infected adults can pass the virus directly to their offspring. The virus does not appear to have an alternate reservoir host, meaning that once infected, the virus relies on amphibians for persistence and spread. Rhinoviruses in fish were first reported in Australia in 1986, where it caused epizootic hematopoietic necrosis in redfin perch. The disease caused extensive tissue death necrosis in the liver, pancreas, and hematopoietic cells of the kidney and spleen. Later, the disease was identified in farm rainbow trout in the same water systems as the affected redfin perch. The virus was experimentally transmitted to 7 additional fish species in Australia. Fingerling and juvenile fish are most commonly affected by the epizootic hematopoietic necrosis, but when the virus is newly introduced to a population, adults can also become susceptible. For the diagnosis, rhinoviruses in fish can be readily detected by isolating the virus from infected internal organs, such as the kidneys, spleen, and liver, using fish origin cell lines. These samples are incubated at 20 to 25 degrees Celsius. The virus can be identified and distinguished from their own rhinoviruses using DNA-based diagnostic procedures. Additional rhinovirus outbreaks have been detected in the cultured freshwater populations of siluriid and ictalurid catfish in Europe, as well as in Atlantic cod fry in a hatchery in Denmark. Largemouth bass virus, also known as Santee Cooper virus, has been associated with large-scale seasonal mortality in wild largemouth bass in the United States. The virus affects multiple internal tissues, including the swim bladder, causing it to become reddened, enlarged with a yellow fluid. This results in the fish floating to the surface, often signaling the first indication of the disease. Similar to amphibians, rhinoviruses can be isolated from asymptomatic fish, which contributes to the unintentional dispersal of the virus through international trade of live fish and amphibians. The transmission between amphibians and fish studies have shown that rhinoviruses can be transmitted between amphibians and fish, both in natural and experimental settings. Rhinoviruses are also increasingly recognized as including the turtles, tortoise, lizards, and snakes. These infections often result in the disease syndrome similar to those seen in amphibians and fish.
Megalocystiviruses and lymphocystiviruses are both members of the Iridoviridae family that affects fish, but they differ in the disease they cause in their effects on host cells. For megalocystiviruses, the host range and impact. These viruses have a significant impact on commercial fish production, affecting both food and ornamental fish. Over 30 species of marine and freshwater fish from Japan, the South China Sea, and Southeast Asia have been identified as potential hosts. Disease characteristics. Megalocystivirus infections can cause high mortality rates, sometimes reaching 100% in captive populations. Diseased fish exhibits lethargy, severe anemia, and hemorrhages in their gills. Sa first picture, napapakita dyan yung lethargy. And the second picture, yung severe anemia. And lastly, yung hemorrhages sa mga gills nila. For the pathology, at necropsy, the spleen may be significantly enlarged. On a microscopic level, there are numerous large basophilic cytomegalic cells in internal organs such as the spleen, kidney, intestine, eye, pancreas, liver, heart, gill, brain, and intestine. These cells contain developing and complete virions and characteristic of these infections. For the diagnosis, unlike ranaviruses, megalocystiviruses are often difficult to isolate cell culture. Historically, diagnosis has relied on histologic evaluation and electron microscopy. However, DNA-based methods like PCR are now commonly used. For the transmission of megalocystiviruses, it is transmitted horizontally among fish in the water, and there's no evidence of vertical transmission, meaning sa horizontal transmission, scale-to-scale contact or physical contact. And yung sa vertical transmission, hindi siya napapasa sa offspring, from parent to offspring. For the control methods include using pathogen-free fish, improved sanitation on fish farms, and husbandry practices that reduce stress. A formalin-killed virus vaccine administered by injection has been effective in controlling Red Sea bream iridovirus in Japan. For lymphocyst viruses, disease and characteristics. Lymphocystis is a benign, self-limiting disease that affects a broad range of freshwater and marine fish. It is characterized by the formation of raised, pearl-like lesions called lymphocysts on the skin and gills. For the pathology, these viruses infect and transform fibroblasts of the skin, gills, and internal connective tissues, causing remarkable hypertrophy, enlargement of affected cells. Lymphocysts can grow up to 100,000 times the normal cell size, which is caused by a virus-mediated arrest of cell division but not cell growth. These cells contain developing virions. The characteristic histologic appearance of lymphocysts is diagnostic for lymphocystis disease, although electron microscopy can be used to confirm the presence of iridovirus virions. Lymphocystis infections are seldom fatal and most often fish recovered by sloughing the external lymphocysts. The main impact is the loss of commercial value due to the cosmetic effects that occur in both cultured and wild-caught fish sold as food. Heavy infections in the oral region may inhibit feeding, and these viral infections can also allow for secondary pathogen entry. For the transmission, it is likely through contact with the virus released from ruptured lymphocysts, meaning it is horizontal transmission. It is scale to scale or skin to skin contact for the fishes. For the control, separation and quarantine of infected fish are used to reduce infections. Genetic analysis suggests that susceptibility to the virus may have a genetic basis.
First is erythrocytic necrosis virus or ENV. ENV is a group of unclassified iridoviruses that affect the red blood cells of various marine fish species, including herring, cod, and some salmonids in the North Pacific and North Atlantic Oceans. So, where is it found? The virus is present in immature blood forming cells or erythrocytes. What does it do? In heavy infections, it causes severe anemia, leading to significant losses in both wild and farmed fish populations. How is it identified? Infected red blood cells develop distinct circular inclusions, visible under a microscope, and electron microscopy confirms the presence of virus particles. Has it been isolated? No, because there are no suitable blood cell lines available for virus culture. How is it transmitted? Experimentally, it has been transmitted through injections of infected RBC, but many aspects of the disease remain uncharacterized. Does it affect other animals? Well, similar viruses have been observed in reptiles and amphibians, but their connection to fish viruses remains unclear. So here in this slide, it shows the pale gills of a herring infected with viral erythrocytic necrosis or VN. So in the top part, the top fish, it indicates anemia, which means the fish has a lower number of red blood cells. So VN is caused by an iridovirus, which is ENV, that destroys red blood cells. reducing their oxygen carrying capacity of the blood. This leads to pale or whitish gills instead of the normal red color, which is seen in the healthy herring at the bottom. The healthy fish has sufficient red blood cells, allowing its gills to appear vibrant and rich in color. Next, we have white sturgeon iridovirus or WSIV. So WSIV is another unclassified iridovirus that was first recognized in the 1980s in California as the cause of disease outbreaks in farmed juvenile white sturgeon. So what does it do? The virus attacks the skin and gills, leading to tissue destruction, which affects the fish's respiration and osmotic balance. Why is it important? WSIV is the most problematic viral disease in white sturgeon farmed for meat and caviar. Where has it spread? Originally found in the Pacific Northwest, it has since been detected in wild and captive white sturgeon worldwide due to the export of live fish. How is it identified? So, infected fish show enlarged discolored cells in the skin and gill epithelium, often leading to tissue necrosis. In electron microscopy, it reveals virus particles, and more recently, PCR tests have been developed for confirmation. How is it transmitted? First, through contaminated water, meaning that infected fish can spread it easily in aquaculture. Second, vertically through gametes. Infected adult fish can pass the virus to their offspring. In control measures, first, by separating different age groups of sturgeon. Second, by isolating infected fish to prevent further spread. So, in conclusion, both of these iridoviruses pose serious threats to fish populations, whether in the wild or in aquaculture. Since there are no vaccines, early detection, strict biosecurity, and proper management are essential in controlling their impact. As shown in the picture, the A picture, the redness around the mouth, occurs due to inflammation and hemorrhaging caused by the virus. The infection damages blood vessels, leading to leakage of blood into the surrounding tissues, making the area appear red. In the letter B picture, increase gill volume. The gills become swollen due to inflammation and hyperplasia, or abnormal cell growth, as the fish's immune system responds to the infection. This swelling can make breathing difficult and reduce oxygen exchange efficiency. Lastly, I'll be discussing iridoviruses in mollusks, particularly their impact on oysters. These viruses have been linked to mass mortality events in both larval and adult oysters in Europe and North America. Iridoviruses in adult oysters. So, in the early 1970s, the Portuguese oyster industry along France's Atlantic coast suffered catastrophic losses due to an iridovirus. The virus caused severe gill necrosis and infected hemocytes or immune cells in oysters. Then a second outbreak occurred in 1977, this time affecting Pacific oysters, raising concerns that the introduced species may have spread the virus to local oyster populations. Last for this topic is the oyster velar virus disease. This first identified in the late 1970s in Washington state hatcheries. It caused near 100% mortality in larval Pacific oysters. The virus targets the velum, a ciliated structure that helps larvae swim and feed. The infection leads to blisters, tissue sloughing, and death. The virus is morphologically similar to the one affecting adult oysters in France, but is slightly smaller, specifically 228 nanometers in diameter. So, control measures should be done in early detection of infected oysters, immediate destruction of affected groups, having strict disinfection, especially in hatcheries, to prevent further spread. In conclusion, iridoviruses pose a serious threat to oyster populations, impacting both wild stocks and aquaculture. Since no specific treatment exists again, Strict biosecurity and early intervention are the best ways to prevent major losses.
For our case report on the family Asfarviridae, the ASF outbreak on a Chinese pig farm. The farm, which was established 3 years ago, experienced a rapid escalation in morbidity and mortality among its pigs in August 2018. The outbreak began on August 15, 2018, with several pigs in one of the breeder sheds displaying clinical signs of disease. Within few days, the situation escalated rapidly. August 15 to 2018, 15 to 19, over just 4 days, the farm saw rapid increases in morbidity and mortality. By August 19, 615 pigs were sick and 88 had died. Affected pigs shown signs like high fever from 41 degrees Celsius to 42 degrees Celsius, depression, bloody nasal discharge, diarrhea, respiratory distress, jaundice, and petechial hemorrhages on the skin. On the ASF's diagnosis, the autopsies revealed massive spleen enlargement or splenomegaly, hemorrhages in lymph nodes, and frothy, bloody fluid in the trachea and bronchi. The clinical signs, along with the distinctive findings on autopsy, pointed to African swine fever. For the pathogenesis and spread of African swine fever virus, the ASF virus is responsible for this outbreak is a large, complex DNA virus. It is highly resilient with mechanisms that evade the immune response of infected pigs. For the geographic context, the farm is located in an area with direct land border connections to Russia, where ASF outbreaks have been prevalent. The strain of ASF identified in China is genetically identical to the Georgia strain, which had spread from farms near Russian Black Sea ports. The African swine fever is primarily spread via direct contact with infected pigs, including wild boars, contaminated materials such as feed, equipment, and vehicles used in pig transport. Lastly is the pork products for meat or offal, which may be introduced into farms via trade routes. For the ASF treatment, unfortunately, there is no effective vaccine has yet been developed or approved. Early efforts at creating killed or subunit vaccines have proven ineffective, while attempts to create live attenuated vaccines have faced safety challenges. But for now, prevention through biosecurity is the most effective measure. The most effective treatment is culling of affected pigs. This prevents the spread of the disease to other animals and is the primary means of controlling African swine fever outbreaks. Infected herds and potentially at risk animals on nearby farms are culled to stop further transmission.
And now I'll be discussing about the frog virus 3 or FV3 infection in wood frogs, specifically Rana sylvatica, a case affecting amphibian populations in the northeastern United States, particularly in temperate aquatic habitats. To start, frog virus 3 or FV3 is a significant pathogen belonging to the Iridoviridae family, which primarily affects amphibians. Wood frogs are highly susceptible, and outbreaks often occur in breeding ponds where the virus spreads through contaminated water sources. So, how does FV3 infect wood frogs? The virus is then introduced into the environment through contaminated water, likely from infected larvae or adult amphibians. It enters the frog's body through skin exposure, utilizing its cellular receptors to invade host cells. The skin, liver, and gastrointestinal tract are among the first tissues to be affected. Once inside the host, FV3 spreads rapidly, targeting major organs. Number 1. Liver dysfunction. The liver loses its ability to detoxify and metabolize, leading to systemic complications. Number 2. Kidney failure. Damage to the kidneys result in fluid imbalances, worsening the frog's condition. Last, hemorrhaging. The virus weakens blood vessels, particularly in the lungs and heart, causing internal bleeding. In the most critically affected frogs, multi-organ failure occurs, leading to death. The severity of the disease depends on viral load, environmental factors, and the frog's overall health. Currently, there is no direct antiviral treatment for FV3, but supportive care plays a crucial role. First, clean water systems. Maintaining good water quality helps reduce viral load. Second, temperature management. Adjusting microclimates can help control disease progression. Third and last, quarantine and isolation. Limiting the movement of infected frogs prevents further spread. Since no approved antiviral drugs exist for FB3, prevention focuses on strengthening the frog's resistance and minimizing stress. First, rehydration and nutrition. Since infected frogs tend to eat less and dehydrate, providing clean temperature controlled water and food is essential. Second, stress reduction. Maintaining optimal temperature, humidity, and lighting in captive populations can improve survival rates. Our last slides for this, managing secondary infections. So, FB3 weakens the immune system, making frogs vulnerable to bacterial infections. Although antibiotics won't treat FB3, they can help manage secondary infections. Broad-spectrum antibiotics like enrofloxacin or oxytetracycline may be used when needed. So, to sum up, FB3 is a major threat to wood frog populations with no direct cure available. However, proper habitat management, supportive care, and infection control are keys in minimizing its impact. By understanding FB3 and its effects, we can work toward better conservation efforts for amphibian species. Once again, I'm Kisin Piniscu Valenzuela, and this is our group report about Asfarviridae and Iridoviridae and its case reports. Thank you.